where we'll discuss a little bit about what are the in vitro anti-HIV testing. And when we talk about HIV, what is the progress that we have made? So, what are the recent advances? Because HIV stays one of those, problem that we haven't been able to do anything about. However, a significant progress has been made, so that I guess the quality of life is much better for patients who are suffering from HIV. So, in that context, it becomes very important that we see how we have moved over a period of time, and from the initial diagnosis or initial, testing of HIV, how the essays have been advanced across the years. So, that is what I would be presenting. For the benefit of the students, let me just right from the beginning tell you that I'm going to go over very basic immunology, just brushing up the topics, so that we understand
Are you
why
changing slide, madam?
no, I haven't changed it yet,
Okay,
sir.
okay, fine, fine.
Yeah, so just to give a brief introduction of how the lecture is going to be laid out, we'll just go very briefly over the immunological significance, because if we are talking about HIV, immune system plays a very major role in guiding the HIV principles. So we do that, and we are going to also connect HIV to our immune system, and then we are going to see that how HIV compromises our immune system, leading to a disease called AIDS. And then we are going to discuss what are the different treatment modalities that have been designed to, I won't say cure AIDS, but at least to tackle AIDS. And when we talk about these different treatment modalities, there comes a very prominent point that how do you test the drugs? So, of course, you before you bring any drug to the market, before you can bring it to the patients of HIV, you have to go via the clinical trials. But before you even move on to the clinical trials, of course, you have to do the pre-clinical studies, which will establish the safety as well as the efficacy of the drug. So that is how the lecture is going to be laid out. So before, wasting more time, I'll just get on with uh, what exactly we are concerned about in immunology and what are the HIV basics. So we know this basic thing that we have a immune defense in our body, and this immune defense is against the infection. We also know that the most important part in our immune defense is played by lymphocytes. Lymphocytes are white blood cells. In lymphocytes, also, we have different categories of lymphocytes, and we are going to be focusing more on the CD4 T lymphocytes, because this is the lymphocyte that connects to the HIV infection. And in the context of HIV infection, not only is CD4 T lymphocytes important, we're also going to discuss the number of CD4 T lymphocytes in the blood, which we call it as the CD4 cell count. Why? Because this CD4 cell count is very important for monitoring the HIV infection, how HIV infection is developing over a period of time. It would also give you an estimation of the risk which would arise because of the HIV infection, and that would relate to the illnesses, especially when we talk about AIDS. Based on the cell count, of course, different treatment modalities are, advised for the patients. So, what would be the recommendation? And of course, this is also an indicator, so endpoint indicator that would also tell us how successful our treatment is. So, the effectivity of our treatment is something. Coming to HIV and AIDS, I hope, all the students have this distinction very clear that HIV is a viral infection, and this HIV infection slowly develops into what we call is an HIV disease. And this HIV disease endpoint is actually the AIDS. So it is not right at the onset that you will get AIDS. You will get AIDS only towards the end, when you have a full-fledged HIV disease. So AIDS is the late phase of the HIV disease. And just to familiarize those students who have no idea about what AIDS is, it's an acquired immune deficiency syndrome. Now, why do we have A, I, D, and S? Because each alphabet signifies or would tell you a little bit more about what the disease is. So AIDS, because it is largely an infection. So when we say acquired, it is an acquired infection by, of course, human beings. I, because it is immuno, so it is relates to the immune system. We are calling it as D because it is damaging the function of the immune system. And S syndrome, why? Because it is a collection of many different kind of infections, and there are many illnesses that occur in an organized in the organism. Why? Because we are talking about the immune system, and because our immune system is compromised, it becomes a ready platform for all opportunistic infections to attack us. And that is why we have this as a collection of diseases, and that is what makes it an AIDS. Talking about the immune system, we know that it is protecting our body against the infection. But unlike other immune systems, like for example, you have circulatory system, which is a very defined system. You have respiratory system. Again, it is a very defined in terms of its anatomical structure. You can actually pinpoint, where exactly is your circulatory system, where exactly is your respiratory system. But unlike these defined body system, when we talk about the immune system, it is a very diffused kind of a system, because most of the system is in a dynamic state. Most of it are specific cells which are kind of moving around in your entire blood and the lymph system, so part of your lymphatic system. When we talk about this immune system, where are we getting these cells from our immune system? So, of course, they are developing in the bone marrow. And the stem cells which are present in your bone marrow are the ones, if you all remember the basic biology, that these are the stem cells which are bifurcating into the development of red blood cells, and they're also kind of making the white blood cells. And of course, white blood cells are the ones which we are going to focus on, and white blood cells are also the ones which we call lymphocytes. Just before I move on, it's a very open kind of a discussion platform, so you don't have to wait till the end to ask me to repeat anything or ask any questions. If you have any queries, you can ask me to stop and I'll answer there and then. All right. So we talked about that we are not going to be focusing much on RBCs. We are more concerned about the white blood cells, which are the lymphocytes. When we are talking about the lymphocytes, we all have studied that there are two subpopulations of lymphocytes. We have B lymphocytes and we have T lymphocytes. And in addition to that, you have various other killer lymphocytes. And we are also aware of the basic immune system, where you have antigens, which are the foreign substances which are present either on viruses or on bacteria. And these are the ones which are activating your immune system, and they are the ones which are triggering a reaction. And that is where your immune response comes alive, right? And antigen is a word we all know is used for these infectious agents, which are the ones which are being presented either by the viruses or by the bacteria, and they are the ones which are recognized by your body, that is our immune system. Now, who is recognizing and who is uh, mounting a defense reaction against these antigens are, of course, antibodies. We all know about that. And antibodies are also prepared, we know that they are made by the beta lymphocytes, right? So this is what is called our humoral immune response. So that immune response where the antibodies are coming alive, and they are the ones which are fighting off your antigens. However, now coming to this whole point in the context of HIV infection, these antibodies are not doing much in case of the HIV infection. So, however, so in case of HIV infection, their presence marks or kind of is a diagnostic, outcome, that it will tell you about the presence of HIV in your system. However, they are not mounting a kind of an immune response where they will destroy the HIV antigen. So in this particular case, our humoral immune response, especially in case of HIV infection, is not something that we can really rely on. So now comes the second immune response that we would have studied, and that is the cell-mediated immune response. Now here we have two kind of cells. This is what we are calling the cellular immunity. And these are the two cells which are the T lymphocytes. You have CD4 population and you also have a CD8 kind of cells. Now this is the most important immune response, especially if we have to fight the viruses, and that include all kind of viruses. And for this particular presentation, we are interested in HIV virus, right? Now, CD4 lymphocytes or CD4 cells, we all know that they are called the Th cells or T helper cells, right? They are the ones which are, of course, indispensable, because they are the ones who are recognizing the antigens, who are going to be recognizing your virus, and they are the ones who are going to be presenting them. So, via the antigen presenting cells, they are going to be presenting them for, of course, for mounting a response, and that is like killing the virus. The second category of T lymphocytes is the CD8 T lymphocytes, or which are the CD8 cells. And these CD8 cells are the ones which are going to be actually killing the pathogen. In this case, of course, HIV is what we want to kill. However, as you will see later, that HIV is a very, very, very brainy kind of a virus. And what it does is, instead of attacking any other cell, it is attacking your T cells or CD4 cells. So basically, what it does is it is sabotaging your immune system by entering the T cells and destroying your T cells. And when your T cells are infected, your own body is mounting a response against this, infected T cells, and that is how we lose all our immune, I would say, capability, because the cells which are the ones which are fighting the pathogens, those cells are being killed by the virus. So CD4 cells, as I said already, are very susceptible to the HIV virus, being the most important target for this infection. So of all the cells, when we talk about HIV, it is the CD4 cells that the HIV targets. So what happens to the immune system, when you are being targeted by the HIV? So you know that your CD cells have been, taken over or held hostage by the
So
HIV
one
cells.
student has asked one question. Can you unmute
Yes, sir.
Manmeet Mishra? Can you unmute and speak? Manmeet Mishra, your mic
Yes,
is
yes,
working?
sir, yes, sir.
Yes, please
Yes,
speak.
sir. Ma'am, how does the virus manipulate immune checkpoints to persist in the host?
Uh, could you repeat the question a little slower, because I couldn't make out what you were saying?
How does the virus manipulate immune checkpoints to persist in the host?
How is it manipulating the immune response? So I'm going to come to it later, that how it tricks the system, because so this question is going to be answered when you have an infection. So your virus has taken your CD4 cells hostage. To fight that infection, your body is making more CD4 cells. So it's kind of like, it is catering to the need of the virus, and more CD4 cells are produced. The virus is taking over those CD4 cells as well, and that is how it is kind of like, taking over the entire defense system of your body. But I would be coming to that as well. Did it answer your question? uh, is this what you had asked, or was the question something very different?
Ma'am, something different.
Okay, can you say it again? Are you asking me how it recognizes the CD4
Yes,
cells?
ma'am. How does the virus manipulate immune checkpoints to persist in the host?
Immune checkpoint. How virus manipulate the immune checkpoint?
No, it doesn't. So basically, it is manipulating the immune checkpoints, exactly because. So, okay. So there's something called the critical concentration of the virus in the body. So when you have initial infection of HIV, it is going to just take over few of the CD4 cells, right? Because here you have to understand that it is not antibody mediated response against the virus. Do you understand this point? That in this case, that first checkpoint where you would have antibodies fighting off the virus, that checkpoint has already been made ineffective because it doesn't work via that,
Okay, ma'am. Okay.
right? So what is it doing? It has avoided the humoral system altogether. What it is doing is it is infecting the T cells, right? And
Okay, ma'am. Okay.
Please continue.
it is the infection within the T cells which is initially not going to do anything to your immune system because your immune system is able to fight off that infection. However, they are going to be dividing inside your T cells, and more virus, when that is released in your plasma, in your bloodstream, which is going to keep on taking over the CD4 cells, that is when your immune system gradually is going to be compromised. So it is a very, very, very slow process of HIV infection. It is not like the other viral infection that you get a viral infection and you immediately, like, you have flu, which is like, I mean, it takes 24 hours to develop. HIV in that way is a very, very, very slow infection. And that is where, it manipulates the immune system. I hope now it is better.
Yes,
Yes, ma'am.
yes, yes.
So in case still not, let's just go over a few more slides and see if it answers your question. And then we'll see, I mean, do we need to discuss anything more? Shall I continue, sir?
Yes, yes, madam. Please
All right.
continue.
So this is, I mean, Manpreet, just to answer briefly here as well. So we talked about how HIV is going to be infecting your CD4 cells. So what would be the consequence of that? That the moment your CD cells are infected, your body has recognized the presence of a foreign, let's say virus inside your body. And what are they doing? They are producing more CD4 cells so that they can fight off the newly produced virus, because these viruses are again part of your blood and plasma. So now there are more CD4 cells. However, it is working counterproductive because more CD4 cells mean that the virus have now more cells to infect and reproduce, and it is a snowballing effect where the virus is continuously increasing in number because the CD4 cells are increasing number. So due to the signals from the non-infected, so you have infected CD cells which are releasing the viruses. The release of the virus triggers your immune system to make more CD4 cells, and at the same time the infected CD cells are killing the nearby non-infected CD4 cells. So over a process, like, over a period of time, what you would see is you have more and more infected CD4 cells, and those cells are being uh, destroyed because the virus kills them and releases from them. And over a period of time, we are losing the CD4 cells. So in the process, in the race between the CD4 cells and the HIV, it is the HIV which is slowly winning the race, taking over the CD4 cells. And that is why we do the CD4 cell count of an infected individual. Those patients which have HIV infection, we will see the CD4 count because we know that over a period of time, the CD4 count is going to be decreasing. And of course, when this infection, of course, is going to persist, because once you have an HIV, it has taken over your body and it is going to just wear you out and completely destroy your immune system. And that is what the drug companies are using, the strategy that, uh, you know how can we stop the HIV? We know that we can't do anything much in terms of stopping it. So what we do is we can block either the reproduction of the virus as quickly as possible, because if you know about HIV infection right from before it's too late. Let's say if it is detected very early, it is easy for you to treat the HIV infection, rather than when those cases where it is detected very late, and that's when, like, the virus has taken over your immune system and it becomes very difficult for you to treat it. So as I already said, CD4 count is a very, very, very important marker of your HIV infection. It will tell you about the severity of the HIV infection. So in a normal person, if your CD4 count is somewhere around 600 to 1600, those people who have HIV infection, their CD4 count would go as low as around 50 cells per cubic millimeter if it is not left treated. So you can imagine what happens to your defenses if your CD4 count cells go that low, right? But here is something which are very 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 peculiar about HIV infection, that there are many people. So HIV infection is very individualistic. There are many people who even without drugs are able to control their HIV very well. So probably because they have a very strong immune system. And even given, if it is given a treatment, then you should see a reversal. You should see that your number of CD4 count cells would increase. So that is one of the ways in which you can measure the uh, not only the infection, but also if you are giving some kind of a treatment, how effective the treatment is. That can also be checked via the counting of the CD4 cells. Another thing is, in addition to CD4 cells, we can also count the CD4 percentage. So CD4 percentage is like percentage of CD4 cells in the total population of your, the total immune cells. So that is again another indicator. So those people who do not have HIV, the percentage of CD4 is around 40%, but people with HIV, you can see the percentage can go as low as 12 to 15%. So these are the indicators which will help you keep track of how fast or slow your disease is progressing. Another thing, or another additional, I would say, problem that comes with HIV is infections with the opportunistic microorganism. We already discussed that our CD4 cell count is being compromised, and that is lowering down our defenses. If it is lowering down our immune defenses, we become susceptible to other infections which are not of viral origin. Not every infection has to be a viral origin. So we are prone to all kind of yeast infections. We are prone to all kind of bacterial infections, and those are called the opportunistic infections. And sadly, that is what is the one which we talk when we say about, when we talk about AIDS. That is what the entire AIDS scenario is like, where your immune system is so compromised that your body becomes a target for all kind of infections. So this is all about the immunology. Now let's come back to the HIV virus. So what is an HIV virus? How is it causing an infection? How do you monitor the infection? What has been the course of HIV disease? What are the different drugs that we are currently using? And then how it is related to the viral load. This we have already done, that HIV is human immunodeficiency virus. Immunodeficiency means that we have less immunity. That means our immunity is reduced. They are very small microorganisms. We know that viruses are something which we of course cannot see from naked eyes, and very difficult to see via the normal microscope. We also know that this is one category of pathogens which cannot reproduce without the help of living cells. So they have to grow and multiply only in the living cells. That is why it has been a topic of debate whether they are the living beings or they are the non-living beings. Because if they are not out, if they are not inside your cell, they are as good as dead. And once they are inside the cells, they come alive. So this is something which we have been discussing. uh, when we talk about human immunodeficiency virus, as the name suggests, only human beings are affected by it, and this is one virus that spreads from one human being to another human being. Uh, Additionally, in addition to HIV, those people who have HIV infection, they would also be infected by or co-infected by other viruses. For example, if you have HIV, you are very prone to be infected by hepatitis A, B, and C virus. You can have cytomegalovirus, CMV virus infection, you can also have a herpes virus infection, and it is all logical because we have already discussed that it is all because of the compromised immune system. So if your immune system is down, all kind of viruses, bacteria, yeast, pathogens are going to be attacking your system. So that was all about the HIV. Now let's have a little bit of a look at the HIV structure. If you look at the HIV structure, of course, this is how it would be. But if we are going to have a little bit of an artistic freedom of representing the HIV, few things are very important. Why these things are important? Because all drugs or our understanding of how HIV is manipulating the immune system has come from our understanding of the HIV structure. So if you do not know the HIV structure, it is going to be very difficult for you to understand what kind of treatment can you actually come up against. So few things will stand out. One is this GP. These are the surface glycoproteins. You can see that HIV is an enveloped virus. It has an envelope, and this envelope has certain glycoproteins. The first glycoprotein which you see in this round is the GP120. This is a surface unit. But this surface unit has this transmembrane domain which is GP41. So with GP120 and GP41, this is the units which are used by the virus to infect the CD4 cells. In addition to that, you have this P24. This is a protein 24, which is part of the viral capsid. So you have the capsid, which is capsid is a protective covering which is around the virus nucleic acid. So you can see that the protein is P24. You have this viral nucleic acid, so it has these two RNA strands. Just for those people who do not know, HIV is not a DNA virus. HIV is an RNA virus, and of course, this RNA has to be converted into DNA before the virus can multiply in a eukaryotic system, because we do not have RNA mediated translation, which can start directly from the RNA. We have a DNA mediated machinery. So this RNA has to be converted to a complementary DNA, and that conversion is done by this reverse transcriptase enzyme. So four important things: the presence of surface, antigens, the surface unit 120 and GP41. Then you have this, the capsid protein, which is P24, and then you have reverse transcriptase, the one which is going to be converting your RNA into the DNA. So we have already seen, not going to go over this, but what we will see is how HIV is infecting the cells and what is the life cycle of the HIV. So in this diagram, you will see that this is one host cell which was probably infected by HIV. This cell has been destroyed and all the free HIV has been released from this cell. Now these HIV which are present in your blood, your plasma, are free to infect other CD4 positive cells, and this is precisely what is happening. So first would be the attachment of the virus to your cell membrane. So you have this lymphocyte. On this lymphocyte, the HIV is recognized by the CD4 receptor. Please do understand that this is the CD4 cells which the HIV is infecting. So they are going to be entering via the CD4 receptor. However, once the HIV binds to the CD4 cell, there is a conformational change, and it realigns and binds with the co-receptors which are present on the CD4 cells, which are the CXCR4 and CCR5 receptors. So depending on whether the viruses uses CXCR4 as a co-receptor or CCR5 as a co-receptor, this is called the tropism of the viruses. Many viruses, they prefer the CXCR4 receptors. Many viruses, they prefer the CCR5 receptors, and that is how the virus category, so their nomenclature is defined. Once they are recognized by the receptor, they are going to be now internalized by the cell. You can see how the envelope protein of the virus has fused with the host cell membrane, and there is a release of the matrix protein. The HIV core is released. You can see the release of HIV RNA. Once you have the HIV RNA released, now comes the part where the host cell plays a role, in the transmission of HIV is now this HIV RNA. HIV is taking very intelligently its own one enzyme, which is reverse transcriptase, which would now help the HIV RNA to get converted into the HIV DNA. And this is the DNA that is important, because this is the DNA that the host cell recognizes as a nucleic material, and that is what is going to get integrated into the. So you can see it now from cytoplasm via the nuclear pore complex going inside your nucleus, and this is where it is going to now integrate. You can see this red portion where it is integrated with our genome, our DNA, especially when I say oh, our, it means the people who are infected with the HIV virus, and it is part of your DNA now. And this DNA, of course, now is going through our central dogma, the linear flow of information, where the DNA is going to be converted into RNA. RNA is going to now move to the cytoplasm, and this RNA, because it is the viral RNA, it is now going to synthesize the viral protein. So you have two things. You have synthesis of viral proteins, you have synthesis of new viral DNA. These new viral proteins, and especially if you recognize, if you remember, I talked about the P24. These are the ones which are going to be now packing the virus. So you can see here how the virus is now getting ready to be packed. So it is again going to bud off your own CD4 cells membrane, and the virus assembly is taking place, and it is budding off, and you have a release of a mature HIV virus, again packed inside this whole beautiful system and ready to infect another CD4 cell. Any queries or any questions still here?
They will ask if they find some question, or after finishing your lecture, they will ask.
All right, so
Yes.
that should be fine. So just very briefly, so we know that HIV is a very difficult virus to catch, and we also know that even from one single exposure, you can become HIV positive, because it is all related to. So HIV infection would be at the highest when the virus load in the infected person's blood is the highest, because if the viral load is the highest, that is when it is that person is at its most infectious state, and that is when you have more chances of getting the virus. And that is why monitoring of the viral load is, of course, very, very, very important. Now, when we talk about the infection, as I said, it is a very individualistic virus. It can take, from few years to develop the virus. So those who have gotten the virus, exposure, they may not develop the infection immediately, but over a period of time, as it says, some the treatment. So you can see that how this infection can be dormant for a very long period of time. So dormant doesn't mean that the HIV is not doing anything. It is progressively, increasing in number, and that is why this is something which is a perpetual infection that you would have in your body. But what it also, say here is that just because an individual has not shown the disease or an infection doesn't mean that individual is not infectious. So even without knowing, a person who may not know that he is HIV positive can still infect other people, because he has the virus inside its body all the time. And that is why it is very important that those kind of HIV testings are done regularly, especially those people whom we know could be the ones who have come in contact with the virus. So we have already discussed the viral load test. How the viral load test is done is that you can also check the HIV genetic material, which is the RNA copies. So you do a polymerase chain reaction, and you can check the number of HIV RNA copies in the 1 millimeter of plasma, and that would the measure of also the viral load. So as I said, viral load can be calculated by the CD4 cell count, or you can also calculate the viral load by checking the copies of the viral RNA inside the system. In addition to this, what you are checking, so when you are checking the viral load and CD4 count or mRNA, what you're doing is you're checking it only in the blood system. But you have to also understand this, that this virus is not restricted only to your body fluid in the terms that CD4 cells or your blood plasma. It is also moving all around your system, and there are part of your genital tract or the central nervous system which can become the sanctuary for this virus. So this is one of the reason again that, unless and you can do a checking of all compartments of your body where the virus can take a sanctuary, it becomes very difficult for you to treat the virus because the infection will keep on coming back. And this is something where the virus can develop a drug resistance, because it is developing very differently in each and every one of your compartment. So if you check the presence of virus in your blood, that is not actually an indicator that this is the same thing that is happening in the entire compartment of your body. So once again, it is a very misleading, and that is what makes this disease very complicated and also difficult to treat, right? Uh, Another thing is mutability of the viruses. That those people who are HIV positive but have not received the treatment, where the reproduction is going at an exponential rate, when anything is dividing at a rate, unnatural rate, of course there are going to be a lot of replication related mistakes. There are a lot of wrong integration of nucleic acid, nucleotides in the DNA, and these mistakes are the ones which will make the genome of the HIV highly mutational. And that is why again it is a problem, because it becomes very difficult for you to come up with a drug or a vaccine, especially a vaccine against virus, because by the time you develop the vaccine, the virus would have mutated and that vaccine will become ineffective. So again, mutation is one of the attached problem, not just HIV but a mutable HIV, and on top of that, mutations which are drug resistance. So once again, why all these things are important? Because these is the entire premise that we work on, that before that kind of a situation is reached, we should try to get the HIV load to come down, and that can only be done in the early, if the detection of the HIV virus is early. So generally, as it is relating to the CD4 cells, agar CD4 cells are count is very high in your body, that means the viral load is low, and CD4 counts is low, it will be the indicator that the viral load is high. So that was as far as the your immunology, your immune system, and how HIV is sabotaging your immune system goes. Now, have we done something about it? Yes, of course. Since there is no vaccine or cure for HIV or AIDS, uh, but it doesn't mean that we cannot improve the quality of life of the patients, or we cannot control the disease. So we can, of course, reduce the level of the infection inside the body, even if we cannot permanently, get rid of that infection. So towards that, we have many kind of drugs which are available in the market. The categories of these drugs are, they are the reverse transcriptase inhibitors. So some of them are direct inhibitors. I'm sure you would have about the NTPs. So when you have nucleotide triphosphates, these are the ones which are coming and elongating your DNA, the complementary strand. So if you are going to make these NTPs, the nucleotide NTPs, where you remove their 3' OH group, uh, you can connect it to the Sanger sequencing of DNA, where if you do not have the 3' OH, that chain of DNA is going to terminate at that point itself. So you have nucleoside transcriptase inhibitors, you have non-nucleoside reverse transcriptase inhibitors, you have nucleotide reverse transcriptase inhibitors. So when you look at the nucleoside and the nucleotide reverse transcriptase inhibitors, they are working as the competitive inhibitors, And the non-nucleoside reverse transcriptase inhibitors, they do not directly integrate into your DNA, but they are the ones which will change the shape of, or the size, or the structure of the enzyme in such a way that the enzyme reverse transcriptase will not be able to work, and that is why they are called the non-competitive inhibitors. Another category is the protease inhibitors. If you remember the life cycle of the HIV virus, we saw how you have proteases, the enzymes which are going to be important for making the capsid of the virus. Capsid is entirely made up of proteins. So if you can have those inhibitors which are not going to let the capsids assemble, then if there is no viral assembly, you are blocking again the infection of the virus. And then in addition to that, there are also entry inhibitors. So you can see that reverse transcriptase inhibitors and protease inhibitors will work only on the virus which is inside the cell. How about that we block those surface receptors, so that the HIV virus will not even enter the cell? For that, you have also the entry inhibitors. However, individually, each category of drugs have been shown to be ineffective. So what is the strategy that we have come up with? We have come up with a strategy that we have developed something which is called HAART, highly active antiretroviral therapy. So it can also be called ART, which is antiretroviral therapy, or CART, which is complete antiretroviral therapy. What does HRT have? The heart is a cocktail of all these things together. So what you do is, in a patient, based on the indication, whether at what stage the patient is, you can give them a combination of a reverse transcriptase inhibitor, you can give them a combination of a protease inhibitor, and you can give them also the entry inhibitors. So this has been one of the strategies. So this is something where once a person is infected and have gone beyond a critical load where you need to treat the HIV, and it is going to be developing into AIDS, these are the drugs that can be given to the patient. However, uh, we are also going to tackle another scenario, and that scenario is, what if we do not even get an HIV infection? What if we can have a preventive measure that we can, we know that once it is inside our body, we can't get rid of it. So, can we do something where we are protected against the HIV infection? And that is where, of course, we have another category of microbicides. So till now, what we have discussed is an infection and how to fight off that infection. But now we are talking about how to prevent getting infected by HIV virus, and that is where microbicides play a role. Now, what are these microbicides? As the name suggests, it is microbe and sites. Cidal means anything which is going to kill the microorganism. So microbicide can be a gel, it can be a cream, it can be a sponge, it can be a suppository, or it can be a film that can be, that can be used for vaginal application, that can be used for rectal application, so that you have very, very, very reduced chance of getting the sexually transmitted disease. Either it can be HIV, it can be chlamydia, it can be gonorrhea, or it can be HPV, or other sexually transmitted disease. So this is what is the preventive one, that before sexual intercourse, you can apply this cream or a gel, or you can use the suppository, and you can insert it in your vagina or your rectum. So, who all can use microbicide? Of course, microbicide can be used by women to protect themselves. You all know that the women who are in I would say in the area of flesh trade, they are the ones which are more vulnerable. So those women can use microbicides to protect themselves. Even men, those who are heterosexual men, or the men having sex, so men having sex with the males, having sex with the males, they can also use the microbicides. HIV plus women can also use microbicide so that they do not infect their partners. Same thing that HIV plus men could also use microbicide so that they do not infect their partners. So microbicide products, although they have been designed for the vaginal use, they can also be used for the anal intercourse, and you can uh, prevent or protect yourself from the HIV infection. So here comes the strategy. As I said, when you have a potential anti-HIV candidate, now the anti-HIV candidate can be your microbicide, it can be your drug. What would be the strategy? The strategy would be, as I
How many
said,
slides, madam, left?
sir, what is the time? I can go over it quickly.
5 minutes. हो
All right,
जाएगा.
great. So I'll go over it quickly. Just give me another 5 minutes extra.
ठीक है
So
मैडम,
you
ठीक
are
है.
going to do the clinical studies, and before you can do the clinical studies, because you cannot put any drug straight away into a person, you have to do the preclinical studies that you will check it either in vitro or you will check it on animals for two purposes. First, that the drug is safe, and also that the drug is effective. Only when you show in the preclinical studies that in a particular dose the drug is safe and it is effective, that you are allowed to move on to the clinical studies. So preclinical studies, of course, is checking the safety and effectiveness of the drug or the ingredient. Will not go over it. Now, what are the in vitro testing models that we have? The in vitro testing models will depend on the kind of drug, whether your drug is an entry or a fusion inhibitor, whether your drug is NRTI or NNRTI, whether your drug is a protease inhibitor. On top of that, it would also depend on the infection mode of the virus, that which cells is the virus infecting and what are the receptors which are involved. As I already said, CD4 cells which are expressing either CXCR4 receptor or CCR5 receptors, which are the chemokine receptors which your body is generally using to fight off infections. Those are the cell testing systems you will use. What kind of in vitro cell lines you can use? You can use the modified HeLa cells, for example, the TZMBL system, and you can also have the natural vital target. For example, you also have a T lymphoid cell line, which is the MT2 cell line. So these are the systems which can be used. I'll very briefly discuss this system, that you have the cell lines, you have T cell lines, you also have the primary isolate, which is like the most infectious virus, unmutated virus, and you can also use this in combination, that you have T cell line, which would be inoculated with the primary isolate. Based on which cells you are going to infect, it can be the CD4 cell line, it can be the peripheral blood mononucleoside, or you can also have the engineered cell lines. You are going to incubate for a few days with the virus, with the drugs and the cell, and at the end of that you are going to have an endpoint measurement, whether you can see how many cells has been killed, if there is a reduction in plaque formation, if there's a reduction in syncytia formation. You can also check for the presence of the GAG antigen, which I said is the one which is making the P24 antigen. You can check the reverse transcriptase activity via ELISA or FACS, or you can do the luciferase assay as an endpoint measurement. You can also have a GFP protein, beta gal, or alkaline phosphatase as the endpoint measurement. So this is how you're going to design the assay, but the endpoint is how much are the target cells viable, what is the P24 antigen level, what is the reverse transcriptase activity, syncytia formation, and immunoprofile. So before going into this, I'll just show you very quickly how you do this assay. So this is a TZMBL cell line in which you have beta galactoside gene and under the luciferase. So luciferase is something which is going to grow. If the drug is inactive, then of course virus is going to bind to beta gal gene, activate it, and you are going to see the fluorescence. If the drug is active, that means if the drug has managed to kill the virus, your beta gal is not going to be activated and you are going to see the lights off. So you have light on if the drug is inactive, and you have light off if the drug is active. So this is one of the system. There are lots of variants of this system. For the paucity of time, I'm not going to go into this. We can have probably another discussion. In addition to TZMBL system, we also have MT2. So TZMBL system was a HeLa cell system, which had been used for evaluation, for endpoint evaluation. But MT2 cell is the lymphocyte-based cell system where you can carry out the same kind of assays for checking the activity of the drug, and you will have an endpoint measurement. In this case, the endpoint is the presence of P24 antigen. If your drug is active, you will not have the virus, so you will not be negative for P24 antigen. If your drug is inactive, of course, your virus will be present and you will have P24 antigen present inside the system. So these are the few tests. Now, in addition to these in vitro tests, since we have a system where the virus is infecting your body or entering your body either through your vagina um, or through your rectum, ex vivo tissue models have been developed which will mimic those environment. And just very quickly, let me go over this. So I'll just show you one of the cervical explant. So this is the cervical explant that has been taken from those women who have gone who had their cervix removed. They have undergone hysterectomy beyond a certain age, and you can see that this is an entire tissue. So with the help of this cutter, what you do is you remove this section and you put it in these kind of cells. So if you, I don't know if it's visible, Gopalji sir, is it visible? Can you see the tissue coming out of this well?
Yes, yes, yes.
So this is a very, very, very recent and developed by the King's College and also the Imperial College of London, where you can have you can see on the upper side is the epithelial surface, and the entire lower layers of the cells, epithelium, the endodermis and the mesodermis are all facing inside. So what you can do is you can put your treatment on the upper level and see if it diffuses inside. So if you can check for those kind of drugs, microbicides, which will prevent the entry. These are specially developed so that we can prevent the entry of virus either through vagina or through rectum, and these are the microbicides. So if we have to develop the microbicides, then these kind of cervical explant systems are the ones which are going to be really handy. These are just few of the pictures. You can see how the cervical explants. In addition to cervical explants, we can also have the colorectal explants, which is from your rectum, especially for those people who are gay, who are bisexual, who are the men having sex with men. For those people, the vaginal system is not going to work. So you can have a colorectal explant system also, through which you can check whether the microbicide that you have developed, it is working or not, whether it is stopping the entry of virus or it is not stopping the entry of virus. So of course, we can see that there are advantages. Vaginal and rectal epithelial cells are involved in HIV transmission. They are the first mucous membrane which are exposed to the virus and the viral product. And if we use this system, we are preserving the tissue architecture and the entire cellular background. So it becomes really, really a good system for checking the microbicide. However, what is the limitation of the system? That availability of the tissue. You have to wait for people to go hysterectomy, and only if you have that, you are going to be able to use the system. So, of course, this tissue explant model is one of the novel model which kind of simulates the in vivo condition. Uh, I will not go into the details of it, but any microbicide that is developed, as I said, virus is not the only infection that can be transmitted via sex. You have other organisms like HSV, chlamydia, candida, Neisseria, and Haemophilus infections that can also be transmitted by the vaginal and the rectal route. So whenever you're testing the microbicide for anti-HIV entry, you can also check whether it is also effective against these infectious organisms. and in that way you would have kind of like all the solution in one gel or one cream. If you can see that one such preparation is able to contain all the infection. So you can check for HSV. There are assays, chlamydia. Then you have anti-STI activities for a group of infections which have Neisseria, Haemophilus, Treponema, Gardnerella, and Candida. All infections can be checked with that. So just to summarize the entire lecture that has happened, we saw how the virus exhibits a tropism. It can either be internalized via CCR4 receptor or it can be internalized via CXCR5 receptor. However, we have developed certain assays with which you can check the efficacy and the safety of the drugs. We saw that we have TZMBL assay. We saw that there was MT1 assays, MT2 assays. We also saw how we can have different kind of products, which can be the microbicides, which are the preventive, I would say intervention, so that we do not get the virus in our system. But at the same time, we have seen that there are drugs which are available once you have virus inside your system that would reduce the viral count, and it would reduce the multiplication of virus inside the system. You have lots of in vitro assays which will check the cytotoxicity, anti-HIV activity, anti-STI activity, which is extremely important. Uh, all this work that I have presented today, I had done it when I was working as a research scientist in National AIDS Research Institute. We had lots of collaborators from NIH, we had collaborators from Conrad USA, we had collaborators from King's College London, collaborators from Imperial College London, and of course I was working in National AIDS Research Institute. So as a disclaimer, none of this work has been completely done by Nari. It is a work from all different labs, and I have taken the material from all these labs.